My name is Jenny Klein. I am the Director of Program Management and Operations at Odylia. So I deal with a lot of our pipeline programs, getting that from preclinical through IND submission. And I will pass this off to Ashley.
Hi, everyone. Ashley Winslow. I'm CEO, CSO for Odylia. I lead the organization as a whole, both from a business and scientific perspective, and a lot of our partnering initiatives. So we'll talk about that a little bit later. And we've included some blurbs about us in the background information. So I'm going to keep it short today, but we will be posting this on YouTube so you guys can follow up. And if you have any questions on anything we present today, feel free to reach out. Thank you. Catherine?
Hi, I'm Catherine. I'm a scientist at Odylia.
Hi, I'm Heather Green, the Director of Development, which in nonprofit world means fundraising.
All right. All right, with that, we're going to go ahead and get started. We've tried to keep things a little bit short today so that we have time for questions and answers at the end, as usual. I think we've been running close on time in previous presentations, so we tried to keep things streamlined. But we did feel like it's an important update today. We usually, I think with the community, have updates 2 to 3 times a year, and I think a lot of progress has been made since our last update in, was that August? September? June.
June. Yep, June of 2024.
All right, thank you. So for those of you who might be new to Odylia, I just wanted to go through a brief primer on Odylia. We are a nonprofit, and that's a 501c3. We are developing gene therapies primarily for rare inherited retinal disorders. RPGRIP1 is our flagship program. It's our lead program. We do have 3 gene therapies for rare inherited retinal disorders in development currently. And our RPGRIP1 program, the other way you might see this referred to, and actually we should include this in our slides going forward. And you may have seen this in communications that have come out as OT-004. That is the same program that we're talking about, have been talking about with you guys. But in the biotech and pharma world, you tend to adopt specific set of combination of letters and numbers at some point with the program. So OT-004 is the same as this program. Here's our lead. We have spoken to you guys in the past about the orphan drug designation we have for the program and the rare pediatric disease designation. We had a big announcement last year with FFB funding, which has been really a boost for the program. And then just to orient you to the other programs, we also have in development gene therapy using the same INC80 capsid that we use for this program. That's the delivery part of the gene therapy vector. We have an USH1C program to treat vision loss in the context of that specific form of Usher syndrome. And then we have a new program that we're hopefully going to roll out that announcement more formally in the next, I would say, month, maybe even a few weeks. And with that, another part of Odylia we haven't, I think, spoken with this group about in much detail is Bridge Solutions. And that's where we take the breadth of learning and expertise that sits in Odylia and our network and try to make that available to the rare disease community as a whole. So Odylia is a rare disease company. We have expertise in the drug development process, but recognizing the challenges for rare diseases across the board, many of those we've seen with our RPGRIP1 program. Many of you are familiar with those. We're trying to leverage those learnings and lessons and make that process easier for patient groups who are navigating that space as well. So this allows us, through our learnings with the RPGRIP1 program, to broaden our impact and help others kind of feed that back to the community. All right, so with that, I think I'm handing this to Jenny.
Thank you. So real quick, we wanted to update you guys on the progress that has been made since our last Q&A session back in June of 2024. So we've been very busy on the manufacturing front and regulatory front since we last spoke to you. We have gone ahead and we've completed a 50-liter scale-up production with our manufacturing partner, who is Andolin Biosciences. So we are still characterizing that batch, but the production itself has been completed to this point. A big milestone for us is we've also gone ahead and prepared our pre-IND meeting package and submitted that meeting package to the FDA. And we had a meeting with them at the end of January. And so we had some great feedback, and Catherine will discuss a little bit more about what that looked like in January. But that was a great step forward for the program overall. We've also initiated steps for our potency assay. So that assay is currently in development, and that assay is important because it will help us measure the activity of our gene therapy. So that has been started and it is ongoing. And then we've also identified our preclinical safety study partner, or in other terms, our toxicology partner. So this is the final preclinical study that will need to be conducted before the first in human clinical trial. And then we've also been exploring additional program funding partners.
So we wanted to give you guys a recap on our pre-IND meeting. So for those of you who don't know, a pre-IND meeting is the first opportunity that the agency has to see our program, to see all of our so far preclinical data and our plans for moving forward. It's also the first time we have to go in front of the agency and present that data to them and get feedback from them. So this meeting really gives that regulatory body a look at a number of things, including the preclinical data to date, our current manufacturing process, and then the plans for a safety study, and then sort of preliminary clinical trial plans. And this is presented to them in a briefing book that we worked on for a long time with the help of the patient advisory board and the clinical advisory board as well. And this pre-IND meeting we had in January, and that's when experts in preclinical testing, in manufacturing, clinical trials, gene therapy, they all come together and discuss our program with us. And then the feedback that we receive from them is then implemented moving forward into our plan. So we received overwhelmingly positive feedback with very minimal changes to our plans moving forward. So that was really exciting. We were really happy with that meeting. For manufacturing, the FDA really approved of, our vector and our plans. On the preclinical front, we had done most of our preclinical testing, and they were very supportive of all the data that was coming from that it supported safety and preliminary efficacy. Our sort of final preclinical data set is the final safety study, and the FDA agreed with our plan for that study. And then on the clinical side, the FDA was supportive of our clinical trial plan, which we were able to generate with, like I said, the help of our patient advisory board and the clinical advisory board. So all of this was really exciting. It's a really important step for Odylia and for this program moving forward, to get that stamp of approval from the FDA in that pre-IND meeting. So we were very happy to have that.
And then as for next steps with this program, now that we've passed that first regulatory hurdle with the pre-IND, we need to go ahead and initiate the vector manufacturing to use in that final preclinical toxicology study. So that is something we will be doing this year. And we also need to continue developing tests that validate our manufacturing process. So that is something that we've been doing and something that will be ongoing throughout this year. And then we will also need to begin developing tests that measure the preclinical safety data. So those tests will be developed specifically for that safety study. And then, of course, we need to go ahead and conduct that final safety study and round out that preclinical data set. So at a more bigger picture, this means like these are all the activities that we really need to do before beginning clinical phase activities. And then at this point, we are looking at the potential clinical trial start for 2027. I will be very brief on the timeline slide. And of course, if you all have questions at the end, we can come back to the slide. But the black dotted line is where we currently are today. Everything in green are the activities we've completed. The activities in yellow are things that are currently ongoing. And then the things in the orange or salmon color are activities that need to be completed. And really, we're at that last phase of the preclinical side of things, which is producing our product for the toxicology study and developing those assays to test those measures in our preclinical study before an IND submission and moving into first-in-human clinical trials.
Jenny, I was thinking we could pause here just to see if there's any questions about the comments from the FDA, the feedback we received, the where the program's been to date, and the plans going forward. So feel free if anyone has any questions to either type them in the chat, or you can also, I think, raise your hand or maybe turn on. I think the best way is maybe raise your hand, which is, depending on your screen, an option. I'm trying to orient where it might be.
I think it's under the React
Yes, which
button at the
I think is
bottom.
different from my screen. Actually, I'm not seeing it usual.
Yeah.
I'll go back. Do we have any questions? All right. If not, we will have plenty of time at the end because we're moving through this actually pretty quickly to come back to questions. All right. Heather?
Well, first, let me say it's great to see so many people on the call today. Some of you are, I feel, old friends and some new folks, and it's nice to see you. We're glad you're here today. We're really excited about how far we've come and all that we've been able to accomplish. And part of that is because of you and the support you've provided and the members of the Patient Advisory Board, some of whom are on the call today. So thank you for all of that. It's hard to believe that we started this back in 2022, and at the time, we had planned to we needed to raise $5 million, which seemed daunting and large. But here we are 3 years later, and we are so close to getting to clinical trials, and it's exciting. So you can see the thermometer there on the right, but well, my right. And the funds that this community and our supporters have donated for the RPGRIP1 program have helped us get to where we are. So all of the things that Jenny mentioned, the milestones and the accomplishments to date, in part, are because of the support that y'all have provided. So it's really, really been important to the program to keep it moving forward. Some of the things that were done last year that were really great. We had a family that did a couple restaurant fundraisers. We also had a family that did, not exactly sure, but it was something with beards, and everyone in the company grew a beard. Well, all the men in the company grew a beard in November, and they raised money, and then their company matched all the money that they raised. So that was a significant boost. And we've also had a family that reached out during the holiday season, and instead of gifts, asked for contributions to be made to the RPGRIP1 program and just what we call peer-to-peer fundraising. We have a platform that we've talked to y'all about before, the every.org, where you can create a page and send it to your friends and family, and they donate through that. And we've had a family do that, and it's been wildly successful to raise some of the funds in 2024 that we received. So those are very very successful, but grassroots fundraising that has worked really well in the last year and a half. So we get this question a lot. How much do we need? And you can see the thermometer from our best estimate. And this is a constantly moving target. The things change. We find strategic partners that work with us as a nonprofit, so our costs are not as high as we estimate. estimate sometimes, but the goal to get to IND has always been about $5 million. And we are at the point now where we need about 1.8 to get that last from the dotted line that Jenny showed to IND, which is is sort of the gateway for clinical trials. So we're working, we've had a great deal of success, and we're working really hard to identify any and all possible sources of funding. I did list on this slide some ideas, some things I mentioned that have been really successful. We also do things like we partner with organizations like Minted. They do Christmas cards and framed gifts and things like that, and they will give a portion of their sales to us. There's there's donor advised funds, which you can, if your family has one of those set up, you can donate through that on our website. And actually, there's lots of ideas also on our website, which we'll put in the chat in a second with a link to the page that has that. But we're here. I'm here to answer any questions you might have about fundraising, to help you with any ideas. We're really hoping that our community, you will come together and assist us with the fundraising. We're also approaching foundations that are interested, companies, anybody we can that will help support the program. But all of us can, working together, every little bit will help. And so we're hoping that we can count on you as well to continue to do all the great things that you've been doing. And certainly let me know if you have any questions or ideas of other ways that we can get to the finish line.
With that, I wanted to touch on the other work we've been doing on trying to find a partner for the program that can help support the uncovered costs that we have. So FFB was a massive help on top of really the fundraising that this community has done directly to keep the program going. And I think we have quite a few new folks, I think, that might not be familiar with the story around where the RPGRIP1 program has been. So it might be worth taking a minute to kind of touch back on the history. It's a history that's not unique in the rare disease space, unfortunately. But the program originally started in the early days of Odylia, before Odylia was founded. RPGRIP1 sat in Luc Vandenberg's lab. And really, with the frustration of not being able to move promising therapeutics forward, Luc got together with Scott Dorfman, our other founder, who comes from the business world, has 2 children with a rare disease. The frustration from both sides that we have promising therapeutics that just aren't moving forward for business reasons. And so that was really the birth of Odylia and the concept. In very early days, we out-licensed this program. We generated interest in it, and it was out-licensed to biotech at the time. And like many other rare disease programs, it quickly it came back to us. So it wasn't shelved, which is great. And that's part of what Odilia's mission really focuses on, is championing these programs that, if they look promising and that the technology makes sense to treat the disorder appropriately, we continue to champion the program and ensure it's not shelved, and find a path forward. It can continue to move forward. So after coming back to us, when that company decided that they no longer wanted to pursue their gene therapy portfolio as a whole, not just our program, we quickly retrenched. And I think that's when a lot of these conversations with this community started in a real way, was working with you all to figure out how are we going to fundraise and bring this program forward, and how are we going to really have that conversation about how to take this gene therapy from its root base. And there's a lot of investment at the time, great data generated when it was in that biotech, but take it to that next level. And that's a lot of what you see on the screen today in a very simplified form, is stepping through these different levels of support that we've had over the years. And so I'd say at the base of that is that nearly $500,000 that this community has raised to ensure that this program can continue to move forward. And that's what this is all about. That was the basis. We feel strongly internally for FFB having confidence to fund this program to that next phase. And so we still have a decent amount to go, but every bit matters. And so what we spent a lot of time actually in the past 4 years, I would say, since that program came back to us, almost 5 years, is finding additional partners that are interested in investing in this program and see the value that we see in it. We haven't talked publicly about a deal that fell through at the very last moment in 2023. Unfortunately, we didn't have permission to speak about it, but we were very excited about a potential partnership that year that did not work out. I think it's sometimes that's how the biopharma landscape operates. I've worked for pharma, I've worked for academic entities, and so there's a lot of volatility. And so a lot of our time is spent vetting partners, finding the right partners, but also ensuring that we continue to move these programs forward when this happens from time to time. And so right now we've been working for the past year on a new partnership. We're very excited about that partnership. It is in process. So again, similar to my mention of 2023, there's some level of uncertainty always. And with our first partner, that plan was put in motion. We had a partnership and they changed course. So all this is to say that, these things can shift. It's a constantly shifting landscape, but we are constantly championing these programs with your support, and we really appreciate that. So I'm very hopeful that we will be able to announce a new partnership imminently. That being said, it's in the works. If you happen to have any understanding of the startup world, this company is in startup phase, so they're building everything out, and that time span can be there's a lot in the marketplace right now. I think everybody recognizes we're seeing a lot of volatility, so we're working with that partner. If you happen to have interest in investments or you know anyone in your network or arena and are interested in discussing that further, we can talk about that in a little bit more detail. But I think this is an exciting phase for us. And once we get to that place of really formalizing that partnership, if everything works out, then we're going to be really excited to talk to this community in more detail about who that is, what that means for the program. Regardless, the program still continues forward. We are, as Jenny laid out, pursuing that development plan. Things are continuing to move in process. We have seen timelines slip for that clinical trial date, unfortunately, just because we fell short of funding last year. But we were able to keep the assay development going and the manufacturing going, just not at the fast clip we had hoped to. So we're hoping that this year we'll continue to not just see that progress, but really start to rapidly catch up on that movement forward. All right, and with that, we will go to, I think, our final slide. We had a few questions from the community ahead of the meeting, and then we're going to open it up as well. So are any other companies using the ANK80 vector? And I see a question now. We'll get to that in just a minute. Hold tight. Don't forget your question. So ANK80 is, for those of you new to this space, ANK80 is our delivery mechanism. So the goal of this therapy is for patients lacking a functional RPGRIP1 gene, we are basically putting that gene, a functional copy of RPGRIP1, into an AAV is what we call that delivery mechanism. Our program is specifically utilizing ANK80. There are 2 other companies that we know of established using ANK80. One, probably the most well-known in clinical trials, clinical phase programs, is a program with Lilly. Lilly acquired Acuus back, I want to say about 2 years ago at this point, don't quote me on that, but they are using the ANK80 vector to deliver a gene into the ear and showing actually really amazing results for hearing loss. So that's a story. If you're interested and not able to find those articles, we're happy to share it. And then Affinia is another company that is using ANK80 vector, actually the capsid in a number of different programs, different organ systems. So that's another space to watch. And then I think we're aware of a few other academic programs. I'd say primarily academic programs. Our second program for USH1C is also on ANK80. All right. Shazia, do you want to unmute and ask a question?
I think it's in the chat.
Oh, it's in the chat. Okay, hold on. Okay, so actually that's that question relates to I think number 2, our next question. Can this gene therapy work in older patients? So the RPGRIP1 community, we have a number of adult patients, and actually we've worked with some of you on our patient advisory board, and it's been incredibly helpful feedback. The current clinical trial, what we call a clinical trial synopsis that we discussed with the FDA, outlines our plans for clinical trials if we are running them today. Now, in my experience, and I think the experience of our team and the advisors we work with is what you put in motion today or what you decide on today. You hope a lot of that is carried through, but ultimately there's reasons that that clinical trial design is going to shift and change in ways that we can't predict today. But we put our best foot forward. You use that as a tool to engage the FDA and get feedback on some of the usual obstacles that would exist for clinical trials. And so our feedback was really good. The current design is slated for that first clinical trial be initially in a population of around adolescents. And so that is between, I think our age range was 10 to 17. Is that correct, Jenny and Catherine?
Yes, that's correct.
And that may be something that changes once we actually get to clinical trials. The rationale for positioning in that age group specifically was around balancing our risk-benefit profile. So with many other gene therapies, I think if you followed that landscape with Luxturna, there was more benefit seen in younger populations than in older populations. We're just now coming up on the 10-year anniversary of Luxturna, and so there's I think a big analysis coming out to look at the effectiveness in a lot of different ways. So we'll know probably a lot more in the next year about that long-term profile for that gene therapy. But we do think this has potential benefit in older populations who have retained photoreceptors. So this gene therapy is delivering that RPGRIP1 into photoreceptors. We know a lot of our adult population has some level of retained photoreceptors. They may not be fully functional. They may be what we call dormant. So we think by putting functional RPGRIP1 back into the photoreceptors, as long as they're still intact, might allow some regain of function. But the hope is with our initial clinical trial to show that within the population, that primarily adolescent population in our current proposal, that we'll be able to study the safety profile, we'll be able to understand the effects in that population better and have more information to figure out how we expand into both a younger population very quickly and an older population. So that's going to be a very rapidly developing area where those two populations may have to have separate protocols and endpoints. And so that targeting that kind of middle adolescence period allowed us to best balance that understanding. All right. I know there's a lot of questions. I am going to go through number 3 real quick, and then we can jump into the questions. Maybe Jenny, Catherine, Heather, you guys can start going through those questions and figure out where we should start. Clinical trial participant selection. Each clinical trial has an inclusion and exclusion criteria. Again, those have been discussed with the FDA through our clinical trial synopsis. Those may change, and those may change because of our better understanding once we get to that point through our safety testing, that next phase Jenny talked about, they may also change because we have a better understanding from other programs that we're watching about how best to execute on the program, because we're looking for ways to facilitate us getting meaningful data back out of a clinical trial and the endpoints we use. We will most likely, it's a rare disease population, so we don't know yet where the trial will be. That may be dependent upon that partner if we have one that I was speaking about before. But almost every rare disease clinical trial I've seen, I think similar with the folks on the phone from the Odylia team, it's usually a global call to patients. So you may have one dosing center. We often see patients flown in for that initial dosing and maybe for follow-up, but a lot of that will be determined once we get closer to the clinical trial. And then right now, this is positioned from what we call a gene replacement standpoint. So it's replacing that RPGRIP1 gene in patients that lack it. And so the profile of this is in patients that have what's called a homozygous or compound heterozygous mutation. So you do need mutations in, everybody has 2 copies of RPGRIP1 in both of those copies. That's the only way for where the program is now to ensure that the vision loss within that patient is attributable to RPGRIP1. We do know there's cases, and I think we're starting to better understand where one vision loss gene may interact with another vision loss gene. So that may be something for further down the road, once we better understand the profile of this treatment and the effectiveness that we begin to tackle. But at least in the outset, this would be for patients with 2 RPGRIP1 mutations. All right, Jenny, do you want to jump in questions?
Yeah, absolutely. I can tackle a few questions if you scroll back up to the timeline slide.
Yeah.
So there was a specific question about the beige arrows. So yes, so the beige, orange, salmon, whatever color you want to call these are activities that we need to complete in order to get to a IND submission. So I can walk through each one, but the 2 biggest arrows really are that 50-liter production, and that is material that needs to be generated to begin our safety study. And you see the black arrow going down to the safety study. So those 2 things need to align with each other. And then everything in between that is assays. So they're assay development. So some of these assays belong to the manufacturing bucket, and then a lot of these assays belong to that preclinical safety study bucket. So there are assays that we need to develop to measure specific things, whether that's how our vector's performing or how our gene therapy reacted in that final safety study to make sure that it is safe and efficacious. So that I think hopefully answers that question. And I know there was another question about the toxicology study and site and then the clinical trial site. So those are 2 different things. So when we're talking about a safety study or a toxicology study, that is going to be at a site like a clinical research organization that has experience working with animals, because this will be the final preclinical, so non-clinical study that needs to happen before entering humans. So where we do that toxicology study will ultimately be different than the place we do the clinical trial. So we will have veterinarians on that safety preclinical study where we will have, medical doctors on the clinical trial site for that study.
And to add more specificity, because I wanted just to touch on that difference, and please feel free to reach out. When I can, I'll put the information for reaching out to our info box or to our emails. If you have questions, we're happy to always get on a phone call and explain, but that safety testing is done in animals. So our previous data building up our understanding and confidence in the effectiveness of this gene therapy has been in mouse models of RPGRIP1 vision loss. The safety testing is primarily done in non-human primate or monkey studies. And so that is the gold standard for de-risking or understanding how, if there's, you tend to go up to a higher dose in monkeys than what you would in humans, but to understand the toxicity, if there is any, and then kind of scale back. So most programs, I'm happy to, again, go into details about how that works, what that looks like. But this allows us to go into the human population in our clinical trials with some confidence that we're at a safer dose. Now, of course, with clinical trials, there is always some risk. So that is something that if you are interested in participating, once we get to that point, there's a lot of information about understanding what that risk profile might look like, what that's looked like from other clinical trials, and what we've done to gain confidence that we're going in at a safe and effective dose in this population. Jenny, do we have which question do we want to jump to next?
I know there's a question around the process and timeline overall. So just to touch on quickly, we're looking at a clinical trial start date of 2027 right now. Our timelines were shifted out last year due to some lack in funding. So we kept everything moving. It was just a little bit slower than we wanted it to be. So that's currently what we're looking at. But if there's any more specific questions on the timeline, please let me know. I'm happy to answer those.
I see a question about how we will select volunteers for the clinical trial. So I think that the best way is to stay in touch with us in a few ways. I'm going to jump to that slide so you guys have that information. Here's a contact information slide, but if following us on social media, we're primarily active through LinkedIn and Instagram, but our newsletter is where that list is where we'll put up updates. I think we have a subset of that newsletter email list that is also RPGRIP1 community members that we're aware of. Heather, feel free to chime in if you have more specific information. But that, if when we get to the phase of trial recruitment, we would put out an announcement and a call. We will also probably work with organizations like Hope and Focus and Foundation Fighting Blindness to reach out to patients through their databases and networks. So we will have more detail once we get closer to that time, but we'll be putting calls out through us and other organizations.
Can y'all give a 30,000-foot view of what happens once we get approval for clinical trials? That was one of the questions, is what happens then? Like, how does that process work?
Yeah, so let me back up on top. Let me layer on top of that, Heather. When we're talking about the amount that I'm going to try to go back to a slide, if my computer will let me. Hold on. It's having it interplay with the chat. So when we're looking at, let me see. So IND submission here, this is the last step before we jump into clinical trials. And usually there's some strategy about how you time that. So the activities we're talking about under this amount of money, the 1.8 million, takes us through the last preclinical phase set of activities. We have started on the clinical trial activities. As we mentioned, we have a clinical advisory board, we have a patient advisory board. We've had a conversation with the FDA about our clinical trial design, recognizing that might change, and some of that may happen in the next few years. We will start to, as we get closer, undertake more activities, but this is where it becomes really important to find a potential funding partner to take it through that next step, because clinical trials are expensive. So the activities that really launch the clinical trial phase would be a GMP, what we call a GMP manufacturing batch. That GMP is a step up from it's a step up in the manufacturing process. It's more refined, higher quality vector production, appropriate for use in humans. So what we're working on now is appropriate for use in animals, and we can keep costs down. But once we look to clinical trials, we'd be stepping up that production. And it's a costly, kind of time-consuming process. So the 1.8 million does not include that. We would look to trigger that GMP batch production of the vector, ideally once we have that funding partner secured for the clinical trial. With that, once we have that GMP batch made, we would submit our IND. So an IND submission, we send a lot of information, a wealth of information to the FDA if that's appropriate for holding the trial in the US. We send it to the FDA for review on a lot of the points we talked about in the pre-IND. So looking at the efficacy data in mice, the safety profile in the monkey studies we'll run, the manufacturing process, and our clinical trial design, and get hopefully their agreement to move forward. So they have, once you submit that, a certain period of time just to give that feedback. And if there's no issues or questions or concerns, which oftentimes something will come up, they'll say, hey, they'll flag, we want a different outcome measure, or we want things done a little bit differently, or we want to look back at, some piece of data and detail. That happens quite often. If not, then we would launch that clinical trial. And so recruitment would start. We'd have ideally a clinical trial site identified at that point, and we'd be able to really hit the ground running. Does that help, Heather? Is that what you were getting at?
I mean, so you find the patients, how long, like what's that timeline when you start recruiting patients, and you do that for a certain number of months, and then clinical trials usually take 3 years, 5 years, non-scientists here, but
no, it can depend. And I think we could go into a separate meeting on this if there's interest from the community, but you have to have a staggered start in your first trial because you're testing that this is the primary aim of a phase. We're slating this to be a phase 1/2 trial. A phase 1 trial really assesses safety. Once you get into phase 2 and 3, you have ideally shown it's safe, and now you're looking at the effect on, in this case, vision. So with a rare disease and with gene therapy, you are often doing your phase 1 trials in patients rather than healthy volunteers. So we have the ability to combine safety and efficacy, and that's why we call it a phase 1/2. But we still have to operate under the principle that we have not tested this in humans before, because this would be the first time in this context. So we stagger patients in a phase 1 trial. So we will test and do in that first injection. And this is currently the design of the trials, to inject in one eye and then wait a number of weeks, look at the data and make sure there's no effects that are concerning. And then we would start the next patient, inject them, and go and on and on. We have this, and again, if you guys are interested, we're happy to go into more detail about the design, but it's an odd time because things usually shift. But we have a different dosing scheme depending on the outcome. So we are starting at a middle dose, or we propose to start at a middle dose and then go up to a higher dose that we think would have more efficacy if the profile looks safe. If we were concerned at all, we could back down into a lower dose. So that's our current thinking and design.
there's a question about I'm sensing stem cells, if they have any impact.
Oh, I think was this the umbilical cord question? Not for this program. So the gene therapy, different from other programs where, trying to think of a similarity, you would source maybe stem cells from the patient themselves to regenerate tissues. This is made in a fully it's a manufactured process. So we don't need any material from the patient or family members. The vector's manufactured in a highly controlled, completely manufactured process that there'd be no input from umbilical or stem cell material.
Ashley, there was a question too about efficacy outcomes specific to RPGRIP1, if that might be different to other types of LCA, maybe different from Luxturna, let's say. Um,
I think there's going to be a lot of mostly similarities. That being said, there is a movement in the vision loss space, and I think FFB, Hope and Focus, there's a lot of patient groups we're trying to change the landscape for outcome measures generally, and I think that's across LCA, retinitis pigmentosa. There's a need to have more effective measures. A lot of these diseases are very slow progressing. And while our hope actually with our program, the feedback we've gotten from the clinical advisory group is they're hopeful there's potential for regain of function, but we won't know until we get into the clinic. But at a base level, we hope for stabilization of function. So the function you have at the point of the AAV delivery, we hope you retain that function and it doesn't drop off. The ideal scenario would also be, again, for those photoreceptors that have gone dormant, that they actually regain, they kind of wake back up and they're fully functioning, and you get more vision back, but we won't know till we're there. In the case of that stabilization in a very slow progressing disease, it can be very hard in the timelines of a clinical trial to measure the effects that the FDA is used to seeing. And so this was a frustration, I think, even with Luxturna and follow-on programs to have more effective measures. And we're looking at some of those. I think it's going to matter. We're going to follow in the footsteps of a lot of other programs that are blazing that trail, and we'll learn from that. And we've started that conversation with the FDA as well about the endpoints we'd like to incorporate. So stay tuned.
There's a question about sort of a ballpark of how much clinical trials will cost. And then also for patients specifically, maybe outside the US or.
Yeah, I mean, usually the clinical trial costs are funded by the funding entity. So the hope is we have a funding partner or sponsor for the program at that clinical phase who would be covering those costs. And that wouldn't fall on the patient community. But I think we have seen some emerging models for rare diseases who don't have that funding, where they kind of work together as a patient group and the patients and funding that themselves. We'd ideally not go that direction. But I think anything's on the table to keep the program moving forward. But I think that's not our plan right now. We're very hopeful and very encouraged that there is interest in this program from companies that would like to move it forward to clinical trials.
I think there's a follow-on question from that. What would that cost look like for the funding partner? It sounds like someone might have a lead on a foundation.
That let's we are in the process of updating our clinical trial costs right now. Actually, what was really helpful getting the feedback from the FDA on our clinical trial design is now we have the ability to take that clinical trial design to the clinical CROs that help you execute. We may or may not use one because this is a rare disease program and we know a lot of the clinicians in the space. All that to be said, let's follow up on that conversation in more detail because we have some numbers, we're updating them. I think we probably could at least get to a ballpark and see if that's possible. All right, what else do we have? We have 8 minutes left.
So we have a question on what's the expectation after receiving the gene therapy? Would vision improve? Are we just looking for it to sort of stabilize, or kind of what is that? And then a follow-up of what's the minimum age requirement for a clinical trial?
So as I mentioned before, that minimum age right now, many times the plan you put in place at the stage we're at is not the exact same plan. There's tweaks by the time you get to actually running the clinical trial. Right now, we have a proposed starting minimum age of 10, and that goes up to 17. Again, that's quite possibly going to change. But we have already started talking to the FDA and laying the groundwork for how we would expand in both directions to a younger population and an older population. And those are likely to look a little bit different from one another. So we're probably going to have to be strategic about how that is executed. But the plan is to move into that broad those different age bands. So we're kind of talking about three now: younger than 10, 10 to 17, and 17 and above. Our hope is to move into all those populations very quickly. And then the expectation. So at a minimum, the aim of the gene therapy is to stabilize visual function that is present at the time of delivery. But we are hopeful from what our clinical advisory group has said, and this takes into consideration the RPGRIP1 biology. It takes into consideration what they have seen with other gene therapies or genetic treatments in the retinal space. We're hopeful that there might be regain of function. The more photoreceptors that have gone dormant and not been lost completely, our thinking is the more potential we have for a regain of function. Usually a regain of function is a much quicker time to see an effect than stabilization. So just stabilizing the function at the point of delivery, while that's incredibly important, and we still actually, when we set out on working with this program, that was the original assumption. It's a longer time to prove efficacy in that group, but we still think it's clinically valuable to the population. Are there other ones we missed? I'm skimming through. Jenny, Catherine, Heather, if you guys see anything, feel free to just jump in and answer.
How long do they typically follow somebody
Yeah.
in clinical trial, or, who's been injected in a clinical trial?
Yeah, so I'd say, so we have a plan right now because it's a safety. The primary concern is proving safety and then its effectiveness. Gene therapy is a one-time delivery. So we have in our current plan a 1-year follow-up, but that doesn't mean we don't continue to follow you. So if you receive an injection, that 1-year follow-up is really critical for our engagement with the FDA to say, hey, this looks really safe. We tried to put as actually a short a period of time so that if the data looked promising, convincing, we were confident in it, we can more quickly move into a broader population. That in the reality of gene therapy, it usually takes a few years to prove that effectiveness. You need a much bigger population followed for a longer period of time. With a slow progressing disorder, it's often a 2 to 3 year follow-up. In gene therapy, we have a short-term follow-up, which is that 1 to 3 years. And then you have a long-term follow-up up to 5 years. And we've seen some gene therapy programs, because it's a one-time and delivery with an expected very, very long-term effect. We've seen programs now go out to 7, 9 years of tracking. So it's not to say that it's going to be that long before we can enroll the next patient or the next trial. It's just we continue to follow up on patients to understand the safety profile and the effectiveness profile. What else do we have?
There's one question about the injection. Is it directly to the eye?
It is. So this program's a subretinal delivery because we're targeting photoreceptors, and the best way to get the vector into photoreceptors, which are a difficult cell to target, unfortunately, is through subretinal delivery. So yes, it's a procedure that is invasive, but is the same procedure that Luxturna's using. It will also, as everything, come with its own set of risks. Any invasive surgery will have its own set of risks that will be outlined in detail in the clinical trial protocol, and also consent forms when we get to that point.
But it's done at a medical facility by a medical professional.
Absolutely. Yes. Yes. The our hope just to, I think, improve the outcome of the trial is we're hopeful to have a single center and a single surgeon administering the first clinical trial vector. The hope is the having everybody dosed, that injection process, that's what we mean by dose, at the same center, delivered by the same retinal surgeon, will allow us to improve the outcomes and the data coming out of that. Now, in practice, if things are successful, that will continue to expand to a larger population, but it will always be delivery by a retinal surgeon.
And one more follow-on question to that was, does it need general anesthesia?
Yes, so this will be, again, invasive, done under anesthesia. There's also a steroid regimen that is often, and we've proposed to follow a similar route of other programs, with both local and systemic steroid administration. That is not for it's somewhat for the procedure, but it's also specific to the gene therapy to help the body not reject the gene therapy or respond to it. And these are details that we can go into once we get closer, or if somebody has an interest in understanding more.
I think we've touched on all of the questions in the chat and
Right.
all of the questions that have come through prior to the call.
Okay. I want to just take the last minute. I'm always really, you guys are a fun group to talk to. We try to rush through the meat of it because there's always a lot of questions. Um, so if there's things we've missed, if there's things that you want to understand in more detail, reach out. The contact information is on the screen. We're very easy to talk to about these things in detail, and there's no silly or stupid questions. So please feel free to reach out with anything. Um, we really appreciate the support, the ongoing support. Honestly, at the basis of that thermometer we are showing, I know it was an absolutely daunting number when we first put it up on the screen the first time we had one of these meetings. But we have chipped away at it, and the fundraising this community has done has been the basis for us being able to continue to champion this program, move it forward, and I think gain the interest and confidence from the funders we've now seen come on board. So I'm really hopeful that we will get to the place that we can announce that next partner. Um, if you guys have any thoughts or input or feedback, please reach out, and we'll keep talking to you through newsletters and updates. But otherwise, we'll look to probably do another meeting sometime in the summer. Hopefully, we'll have more updates. All right. Thank you all.